So what we do here is we say instead of transmitting, instead of an antenna transmitting an electromagnetic wave, we now have a bacteria or a cell that's releasing chemical markers. The receiver, instead of also being an antenna, is receiving those markers. The noise takes a fundamentally different form from the kind of noise that we're familiar with. Firstly, we can have reactions in the space. And secondly, probably the most important source of noise in this medium is the randomness of propagation. So instead of a nice wave, now we have these discrete carriers that move with Brownian motion, and they can get lost. They can also take a random amount of time to move from the transmitter to the receiver. So just very briefly, how exactly would you do this? So this is just to give you some motivating ideas as to exactly where this problem is coming from. Let's say I want to send 1 bit. So all I want to do is send a 0 or a 1. How exactly do I do that with chemicals? So let's say we have these genetically engineered cells, and I'm trying to get them to cooperate, and I just want them to send 1 bit from one cell to the other. We'll call this the transmitter cell. We'll call this the receiver cell. How would this work? Well, there's 3 ideas basically. We can try sending our information in the quantity of molecules. So if we want to send a 0, we release no molecules. If we want to send a 1, we release a whole bunch of molecules. And again, these will propagate randomly. There will be some final decision time at which time we have to say whether or not a 0 or a 1 was transmitted. And by that time, a certain number of those molecules over here will have hit over here. Others might get lost in the medium. So in other words, if we observe at least 1 molecule over here, we can say that a 1 was sent. If we observe none, then either they all got lost or a 0 was sent. Here's another idea. What if when we send a 0, we send a molecule of type A, let's say a calcium ion. And if we send a 1, we release a molecule of type B, let's say a sodium ion. As long as the other cell can distinguish between those two, then all the other cell has to do is figure out which one was sent, and then it can decode the message. Or we can send information in timing. What we can do is, in order to send a 0, if all we have is a reservoir of 1 type of molecule, we can release that molecule now. Or if we want to send a 0, if we want to send a 1, we wait a certain amount of time, and then we send it later. So in other words, the information is encoded in the timing. And then what we do at the receiver is measure the arrival time. So all of these, academically speaking, can be encoded into Shannon's channel model. We can take all of these, break them down abstractly, and try to figure out exactly how they fit into the Shannon model of information transfer. And we can do interesting things with that, at least from the academic side. So, coming back to this question, what can academia tell us about this problem? Again, I'm biased, this is my research, I'm an information theorist by background, but my argument is academia can tell us about the feasibility of this idea. The broad feasibility, sort of the notion of what exactly can be accomplished before we hand it off to industry and say let's work together and build a product. So really the kind of question that I as an information theorist am interested in is what kind of data rates can we expect? In other words, before I take this to Steve and say what exactly can we design here, it would be nice to know order of magnitude, what kind of data rates we're looking at. Is it 1 bit per second? Is it 1,000,000 bits per second? Is it 10 to the minus 6 bits per second? What exactly are we looking at here? So I solved this problem. So hopefully most of the people, this is not unfamiliar to most of the people in this room. Basically, I calculate Shannon's capacity of the molecular communication problem. This is an unsolved problem, I should point out. There are lots of features of this problem that make it extremely difficult, but in my work, what I've done is I've found some bounds. And this is the answer. About 1 bit per second per molecule. So if you take most of the models that I've been talking about, plug them into the Shannon's channel model, and try to plug in some realistic parameters for all of the parameters, the answer you get is probably on the order of plus or minus 1 or 2 orders of magnitude, about 1 bit per second per molecule. That doesn't sound like very much, but actually it's spectacular. How much is 1 million molecules? Not very much, right? So you can get 1 million bits per second in principle if you have 1 million molecules sitting around. And if you want to learn more about the academic research, there's the URL molecularcommunication.ca. .ca because all you have to remember is I'm from Canada. So, what can't academia tell us about this problem? Well, now we come back to the incentive structure. So the problem is the incentive structure in academia rewards incremental change that leads to a paper that gets published. So as long as I can put a paper on my CV, and as long as that paper is in a reasonably prestigious venue, but all the academics in the audience know that it is hard, but not terribly hard, to get a paper into a prestigious venue. As long as I can do that, I can advance my CV, I can get promotions, I can get grants, and so on. This incentive structure rewards a couple of things. It rewards working in small groups on bandwagon-y type problems. And this is the problem that we're starting to see in nano networking. So it's starting to acquire a bit of a cachet in communications research circles, and as a result, we're starting to see these sort of incremental papers produced by small groups. Now, I would argue what we really need here, we need industrial involvement, and what we need are massive demonstration projects, probably with 50 to 100 researchers working on them, that show that, for instance, you can indeed inject a person with nanorobots and they can swim to the knee and do something interesting. That's not a problem that can really be solved by a professor and a couple of grad students. It really needs collaboration across a whole variety of fields. It needs significant investment, and this is not the kind of problem that academia is really set up to solve. What we really need is some way of bringing together industry and academia in order to solve these kinds of problems. Moreover, we need to know, I have my own idea and my own vision for what these kinds of, for what this kind of solution, for what this kind of thing can solve, but there might be other applications. And again, academia is not very good at knowledge translation, so what other kinds of solutions can we find? Here's an example. So this was a surprise. I'm working with some industrial collaborators from Japan, and they realized that the nanorobot idea, that's a little bit, it's not super far future, but it's probably a decade away right now. What's here right now are these. This is what's called a microfluidic lab-on-chip. A lab-on-chip is basically a device that performs chemical reactions in a very small space to do something like the following. So today, if you have some kind of a disease that can be determined from your blood or a urine sample or something like that, you go to a lab, you provide the sample, they send it off somewhere, 2 days later you get your answer. With one of these, all of the reactions take place right on the chip and you get your answer in a matter of a few hours. So it turns out that it's very awkward to combine electronic and chemical components on the same substrate. So if you want to do any kind of decision or control, it turns out that molecular communication is an option here. Do everything in the chemistry. So this is an application that I would have never thought of as an academic, and that would probably take an industrial researcher to determine. So what's standardization doing here? What standardization is doing is bringing together the academics and the researchers. Again, standardization has clout in industry. This is something that industrial people have heard of. Standardization is respected in industry. The Standards Association has a good reputation. And so when you come to an industrial representative and say, I have this great idea, and surrounding it is this framework of standards, what they hear is that it's a serious idea with clout, and the standardization project will reduce the risk in trying to translate that knowledge to an industrial project. So the standardization, I would argue, is keeping nano networking on track. It also reduces the bandwagoning effect. So let's look 5 years ahead, and ideally, in 5 years, nano networking will be on everyone's lips, and it'll be then important, for instance, for any researcher who wants funding to include the word nano networking in a grant. Unless we decide exactly what we mean by nano networking today and make that a standard, there's a real danger that the idea will be diluted, and that everyone will be talking about nano networking, but no one will mean anything by it. So this is another way in which standardization helps. It's keeping us from diluting the idea, it's keeping the terminology meaningful, and making sure that people know what we're talking about when we talk about nano networking, so that when I come to a company and say, I'm working on nano networking, they know exactly what I'm talking about. So, to wrap up my part of the talk, I'm just going to talk a little bit more specifically about nano networking standardization activities. So we have 2 activities underway right now, the IEEE 1906.1 working group. This is led by Dr. Bush as the chair and myself as vice chair. We have probably a dozen very active members and a roster of probably 60 or 70 interested people. And our goal is to firstly produce a definition of nano networking, again, to reduce that bandwagoning dilution effect, to produce a framework, for nano communication systems, and to develop a standard glossary of terms around it. There's also the future directions for nano networking research project. This is looking a little further ahead, and basically we'll produce a vision document that will tell industry, here's where a collection of academics and industrial representatives think this field is headed in the next 5 to 10 years. Composition, we've got academic representatives from universities in about a dozen countries, I think, at this point. We've got industrial representatives from not only big names that you've heard of, IBM, GE, Samsung, but also we have significant representatives from startup companies, from small businesses, and so on. I think that's a really healthy mix. Importantly, we also have representatives from government. We've got at least one representative from DARPA, one from NIH, one from the Canadian Space Agency, and these are important to bring to the table, because when academia and industry really get together, what we want is research funding, and the people that provide that funding are these government agencies, so it's important to have them on board as well. They're also excited about this kind of standards initiative. Again, what's our objective? Standardize languages and methods, promote nano networking. So this was another one of our objectives when we showed up to industry and said, we're working on a standards project. Their ears perked up. And then we said, about nano networking. And then they said, oh, okay, that's interesting, what's that? Whereas if we'd led with, we had this funny idea that sort of brings together biology and communications, I'm sure they would have immediately stopped listening. So what we're really interested in is, in part, taking the clout of the Standards Association and attaching it to this new idea. We want to prevent dilution, prevent this bandwagoning that I talked about. We want to really encourage this large-scale interdisciplinary collaboration, and we have done so. So again, we've got industrial representatives, academic representatives. The academic representatives include biologists, so we actually do probably a first for any IEEE standard. We've got biologists on board to help us with it. Current status, the standards project, our definitions are more or less complete. We're moving on into other phases, and we're expecting the final results, let's say 2014, 2015 kind of timeline. The research project, the vision document that I talked about is in progress, expected around 2013. All of the results will be published by the IEEE Standards Association. So for more information, you can feel free to contact me. I think Dr. Bush's email is on his slides, and there's the website for 1906.1. So thank you very much for your attention.